friends and welcome. Today we shall talk about Archaebacteria. The main objectives of today's deliberation are: number 1, to give a basic idea about Archaebacteria; number 2, to know about the history of Archaebacteria; number 3, to gain knowledge about different characteristics of Archaebacteria; number 4, to know about different types of Archaebacteria; and finally, to understand the uses of Archaebacteria. To begin with, let me give you a brief introduction. Archaebacteria are among the earliest forms of life that appeared on Earth billions of years ago. Archaebacteria are interesting microbes characterized by highly diverse morphology and habitat. They have gained special interest among microbiologists and biotechnologists due to habitation in extreme environments of temperature, acidity, and pressure. Archaebacteria are such microorganisms which are prokaryotic in their cell organization but possess a strikingly different cell chemistry when compared with the bacteria. They are genetically as distantly related to bacteria as both are to eukaryotic organisms. This distinction among the Archaebacteria, bacteria and eukaryotes is so profound that Archaebacteria have been given the rank of a domain with a completely different status in the history of organisms classification. These were not recognized as a distinct form of life from bacteria until 1977, when Woos and Fox proposed the archaea as a third domain of life based on small subunit rRNA, that is ssRRNA sequence cataloging. This represented a profound paradigm shift from the dichotomy of eukaryotic and prokaryotic life forms that had previously existed. Not only does archaeal ssRRNA differ in primary sequence from that of the other two domains, that is bacteria and eukaryota, but also each domain was shown to have specific signatures in this conserved molecule, both in primary sequence and in the secondary or tertiary structure. Woos proposed that life on Earth is made of 3 primary lineages, which he referred to as domains. Number 1, Eubacteria, that is Eu means good or true. Number 2, Archaea, that is Archaea means ancient. And number 3, Eukaryota, that is Karya means nut or kernel. Now let us discuss various characteristics of Archaebacteria. Number 1 is habit. Archaea exist in a broad range of habitats and as a major part of global ecosystems, may contribute up to 20% of Earth's biomass. These prokaryotes often occur in extreme aquatic and terrestrial habitats, which may be anaerobic, hypersaline, or hyperthermophilic. The first discovered archaeans were extremophiles. Indeed, some archaea survive high temperatures, often above 100 degrees Celsius, as found in geysers, black smokers, and oil wells. Other common habitats include very cold habitats and highly saline, acidic, or alkaline water. However, archaea include mesophiles that grow in mild conditions, marshlands, savages, oceans, and soils. Some Archaebacteria have been found as symbionts in animal digestive systems. Number 2 is shape and size. Archaebacteria are morphologically diverse and include spheres, spiral, rods, lobe, plate-shaped, irregular shaped or pleomorphic. They may exist as single cells, as aggregates, or form filaments. The size of Archaebacteria ranges from 1/10 of a micrometer to more than 15 micrometers. Number 3 is cell multiplication. Archaebacteria usually reproduce by an asexual process known as binary fission. During this process, the bacterial DNA replicates. The cell wall pinches off in the center, due to which the organism is divided into 2 new cells. Each cell consists of a copy of circular DNA. Some species can multiply from 1 cell into 2 in as less time as 20 minutes. Some also multiply by budding, fragmentation, etc. Next is cell walls. Some Archaebacteria, such as Thermoplasma species, do not contain a cell wall, whereas most others do contain cell walls. The Archaea have long been known to contain unusual cell wall structures and components. Indeed, one of the first major distinguishing features of Archaea used to separate them from bacteria was the lack of murine in their cell envelopes. The cell wall containing Archaea can stain gram-positive or gram-negative. The cell wall containing Archaebacteria are morphologically almost similar to bacteria, but the chemistry of the cell walls of the two are strikingly different. The Archaebacteria cell wall lack peptidoglycan, which is present in the cell wall of eubacteria. Instead, is composed of pseudomurine, which is made up of a combination of an acetylose aminuronic acid and an acetyl glycosamine. This kind of cell wall makes Archaebacteria immune to the effects of lysozyme, which is an enzyme produced by a host's immune system to attack and disable cell walls of pathogenic bacteria. The ability of Archaea to adapt to extreme environments is assisted by the possession of unique cell wall types, which vary from those containing molecules composed of pseudopeptidoglycan closely resembling bacterial peptidoglycan, to cell walls completely lacking a polysaccharide component. A common wall type is a paracrystalline surface layer, that is S layer, consisting of protein or glycoprotein, generally of hexagonal symmetry. Although S layers are common to all groups of Archaea, the biochemical makeup of S layers among species is very diverse, and in some cases, this layer is too supple to contribute to the stability of the cell. In these cases, the function of S layer is unknown, but it has been proposed that the space between the cytoplasmic membrane and the outer surface of the S layer may fulfill the role of a periplasmic space, reminiscent of gram-negative bacteria. Next is appendages. Archaea, like bacteria, may have a variety of appendages extending from the cell surface. Several appear to be unique to Archaea, while others, such as flagella and pili, appear superficially like organelles in bacteria, but have Archaea unique features. Among these features are the grappling hook appendages found in large abundance on the surface of Euryarchaeon discovered in marshes in Germany. Second are the hollow tubes called cannulae, which connect cells of the hyperthermophile Pyrodictyum abaci. Among the appendages found in Archaea that resemble bacterial counterparts, the best studied are archaeal flagella. These differ fundamentally from their bacterial namesakes with genetic and structural evidence suggesting that archaeal flagella are related to bacterial type 4 pili. organelles that mediate the surface motility called twitching. Next are lipids and cell membranes. Another fundamental trait identified early in the study of the domain Archaea was the presence of ether-linked lipids in the cytoplasmic membrane. Phospholipids in the other two domains consist of linear fatty acids ester linked to a glycerol backbone. Archaeal cytoplasmic membranes are typically composed of diphytenyl glycerol diethers containing phytenyl chains consisting of 20 carbons, which form a lipid bilayer, although in some cases, such as those of some thermoacidophiles, the membranes can consist of diphytenyl diglycerol tetraethers, phytenyl chain of 40 carbons, which span the cytoplasmic membrane in a very stable lipid monolayer. Mixtures of the two types can be found in individual species, and other variations such as cyclopentane containing lipids are also found. Besides these fundamental structural differences, the stereochemistry of archaeal lipids is different from those of both bacteria and eukaryota. In archaea, 2,3-syn glycerol backbones are used, while in the other two domains, 1,2-syn glycerol backbones are employed. The ether linkage is much more resistant than the ester linkage to hydrolysis upon exposure to the extremes of pH and temperature found in many archaeal habitats, and it was originally thought that the unique archaeal lipids were a specific adaptation to extreme environments, before it was realized that these lipids are a defining trait of the entire archaeal domain, regardless of their environmental niche. Next, nutrition and physiology. Some are aerobes, some strict anaerobes, and some facultative anaerobes. Some are chemolithoautotrophs, others chemoorganotrophs, and some can grow by changing from one to the other nutritional mode. The growth temperature varies between mesophilic to the hyperthermophilic. The pH growth range is between very acidic to slightly over neutral. Some are obligate halophiles, yet others are halotolerant. Next, genomes. Their DNA contains a single circular molecule which is compact and tightly wound. No protein is associated with DNA. Since these organisms don't have nuclei, the genetic material floats freely in the cytoplasm. They consist of ribosomal RNA, that is rRNA. The archaebacterial cell may contain plasmids, which are small circular pieces of DNA. The genomes can vary in the GC content of their DNA between 21 to 68%, indicating a marked genotypic diversity. Genomic resistance to thermal denaturation and genomic structural integrity in extreme halophiles is related to high intracellular salt concentrations. Next, RNA polymerase structure. Archaebacteria, like bacteria, possess a single type of RNA polymerases but are complex, consisting of up to 14 subunits, as against only few subunits in bacteria. Archaebacteria RNA polymerases are similar to eukaryotic RNA polymerase II. However, archaebacterial RNA polymerases are insensitive to drugs, rifampin and streptolizidin. Unlike E. coli RNA polymerase, archaeal RNA polymerases are unable to initiate transcription in vitro. This is also seen in eukaryotes, where general transcription factors are required for initiation. Archaeal promoters have an 80 resuctance at -32 to -25 base pairs upstream of the transcriptional start. The consensus Secans resembles a eukaryotic data box. Next, tRNA nucleotide secans. The Archaebacteria lack ribothymin in the common R of transfer RNA, that is tRNA, whereas it is present in most tRNAs in bacteria and eukaryotes. Ribothymin is replaced by pseudouridine or L-methyl pseudouridine in Archaebacteria. Also, they are methionine tRNA met rather than formyl methionine tRNA ep-met as indicated tRNA. Next, coenzymes and cofactors. The methane production mechanism of methanogenic Archaebacteria is unique in the microbial world and involves several novel cofactors and coenzymes not found in any other group of microorganisms. These coenzymes and cofactors identified so far are methanofurin, methanopterin, coenzyme M, coenzyme F420, coenzyme F430. Now, let us talk about major types of Archaebacteria. Number 1, methanogens. Methanogens are the largest group of Archaebacteria characterized by their unique energy metabolism, in which they convert fermentation products to either methane or methane and carbon dioxide. The enzymes used in methanogenesis are very oxygen sensitive, so methanogens are very strict anaerobes, hence produce methane in oxygen-free as well as low redox potential environment. They are found in all types of anaerobic environments and are certainly the most prevalent Archaebacteria in the moderate world. There are many known methanogenic genera. The important ones are Methanobacterium, Methanothermus, Methanococcus, Methanomicrobium, Methanogenium, Methanospirillum, and Methanosarcina. This capability of methanogens is commercially exploited in the production of methane and fuel gas inside gobar gas plants. For example, Methanobacterium, Methanococcus. Some of the methanogen Archaebacteria live as symbionts. For example, Methanobacterium inside rumen or first chamber in the stomach of herbivorous animals that chew their cud. That's ruminants, for example, cow, buffalo. These Archaebacteria are helpful to the ruminants in fermentation of cellulose. Some methanogens can grow as autotrophs and utilize carbon dioxide as a carbon source and hydrogen as an energy source. But they may also utilize formate a carbon compound as carbon source. Besides, Methanothrix uses acetate and methanococcides utilize methylamine and methanol. Most known methanogens are mesophilic. That's they require optimum temperatures from 21 degrees Celsius to 45 degrees Celsius. Some are thermophilic. That's they require high temperatures of an optimum between 50 degrees Celsius to 80 degrees Celsius. Now, halophiles. Halophiles are named so because they usually occur in salt-rich substrata like salt pans, salt beds, and salt marshes. They live in extremely salty conditions such as those found in the Great Salt Lake and the Dead Sea. Such environments present two challenges. First, the difference in salt concentration inside and outside the cell is tremendous, creating huge osmotic pressure. While other organisms would rapidly lose all of their water and die, halophiles have adapted to survive within such a difference in water gradient. Second, the salty environments are very alkaline, some having a pH of up to 11.5. Beyond simply surviving within these inhospitable environments, halophiles have incorporated the conditions into their unique photosynthetic pathway. They are aerobic chemoheterotrophs. It is considered that they require approximately 17 to 23% NaCl for good health. For example, Halobacterium, Halococcus. The halophiles are poorly flagellated rods, for example, Halobacterium, ranging up to 8 micrometers in length, and immotile cocci, for example, Halococcus. The Halobacterium species are pleomorphic and can undergo morphological changes in response to variation in the salt content of the environment. A very high salt concentration may cause the cells to become round and ultimately lyse. The halophiles carry on unique version of energy synthesis, namely light-mediated ATP synthesis, which does not involve photosynthesis. The best studied is the Halobacterium selenarium. When subjected to low oxygen concentrations and high light intensity, it synthesizes a modified cell membrane, namely purple membrane, which contains a protein pigment called bacteriorhodopsin. Now, extreme thermophiles. These bacteria live at extremely high temperatures, such as in hot springs, and can also be associated with extreme acid environments. Many depend on sulfur in their metabolism, and many produce sulfuric acid as an end product. Regardless of varying environmental conditions, the ability of thermophiles to thrive in extremely hot environments lies in extremozymes, enzymes geared to work in extremely high temperatures. The amino acids of these extremozymes have special tricks to retain their twisted and folded 3D structures in high heat, where other enzymes would unfold and no longer work. Pyrodictyum is the important example of Archaebacteria that grow in extreme thermophilic conditions, particularly the submarine volcanic habitats such as thermal springs and deep sea hydrothermal vents. This Archaebacterium has been isolated from geothermally heated sea floors and grow at 82 to 110 degrees Celsius temperature. Now, thermoacidophiles. These are ecologically and economically important group of microorganisms which thrive in acidic environments. Almost all thermoacidophiles are obligate anaerobes. The thermoacidophiles are distinguished by their ability to grow in harsher environments of high temperature and low pH value. Basically, these Archaebacteria are chemosynthetic, that is, they obtain energy for synthesis of food from oxidizing sulfur. Under aerobic conditions, they usually oxidize sulfur to sulfuric acid. If the conditions are anaerobic, the thermoacidophiles may reduce sulfur to hydrogen sulfide. Bicarbonates are also precipitated into their carbonate form by their activity. They represent a heterogeneous group which can be sulfolobous, thermoplasma and ferroplasma. Sulfolobus is a gram-negative facultative chemoautotroph. Its cells have an irregular spherical lobed shape. The temperature optimum required is between 70 to 80 degrees Celsius with a pH optimum of between 2 to 3. Sulfolobus grows in hot acidic springs and hot acidic soils all over the world. The main growth substrate of these is the elemental sulfur. Sulfolobus grows on the surface of droplets or crystals of sulfur and oxidizes it to sulfuric acid, which is largely responsible for the acidity of its habitats. Thermoplasma, like mycoplasmas, lacks cell wall. It is a facultative anaerobe which utilizes some mono and disaccharides as source of carbon. Thermoplasma has been found growing only in the refuse piles from coal mines, which contain residual coal and substantial amounts of iron pyrite. Oxidation of iron pyrite by some chemioautotrophic bacteria acidifies and heats the pile, creating a favorable growth environment for thermoplasma, that is temperature of 37 to 65 degrees Celsius having pH 0.5 to 4.5. Ferroplasma is a chemolithotropic relative of thermoplasma. Like thermoplasma, it lacks cell wall and enjoys acidophilic lifestyle, but is not a thermophile as it grows optimally at 35 degrees Celsius in mine tailings containing pyrite. The latter is used as energy source. Now, coming to the uses of Archaebacteria. Extremophile archaea, particularly those resistant either to heat or to extremes of acidity and alkalinity, are a source of enzymes that function under these harsh conditions. These enzymes have found many uses. For example, thermostable DNA polymerases such as the PFU DNA polymerase from Pyrococcus furiosus revolutionized molecular biology by allowing the polymerase chain reaction to be used in research as a simple and rapid technique for cloning DNA. In industry, amylases, galactosidases, and pullulonases in other species of Pyrococcus that function at over 100 degrees Celsius allow food processing at high temperatures, such as the production of low lactose milk and whey. Enzymes from these thermophilic archaea also tend to be very stable in organic solvents, allowing their use in environmentally friendly processes in green chemistry that synthesize organic compounds. This stability makes them easier to use in structural biology. Consequently, the counterparts of bacterial or eukaryotic enzymes from extremophile archaea are often used in structural studies. In contrast to the range of applications of archaean enzymes, the use of the organisms themselves in biotechnology is less developed. Methanogenic archaea are a vital part of sewage treatment since they are part of the community of microorganisms that carry out anaerobic digestion and produce biogas. In mineral processing, acidophilic archaea display promise for the extraction of metals from ores including gold, cobalt, and copper. Archaea host a new class of potentially useful antibiotics. A few of these archaeocins have been characterized, but hundreds more are believed to exist, especially within Haloarchaea and Sulfolobus. These compounds differ in structure from bacterial antibiotics, so they may have novel modes of action. In addition, they may allow the creation of new selectable markers for use in archaeal molecular biology. With this, we come to the end of today's lecture. Hope you have understood it well. Thank you.